Next, I would like to welcome Drs. Molly Gasperini and Eric Jones here from Octant, which is in the San Francisco Bay Area. Octant is a synthetic biology company that uses multiplex methods to study GPCRs for drug design. And additionally, we want to thank them again for sponsoring the poster awards that we received yesterday. Molly, who is now a full scientist at Octant, previously did a graduate work in the lab of Dr. Jay Shanduri, developing CRISPR perturbation screen and other multiplex functional assays. And Dr. Eric Jones, prior to Octant, got his degree from UCLA under the supervision of Sri Gasperini, focused on developing multiplex assays for decoding receptor ligand interactions, which some of the work spurred the Octant company. So Molly and Eric will be talking about selection methods today. So thank you.
All right, hi everyone. I'm excited to talk to you guys about functional selection methods. And more particularly, we hope that you get out of this a survey of the options out there, as well as the relative advantages and disadvantages of the different approaches.
Click that.
Okay.
Hey.
Okay, so functional selection is the crux of every DMS experiment. Doesn't matter how balanced your library is, how much coverage of your variants you have, if you found the perfect expression host, but if you don't have a way to robustly functionally stratify your variants, then what is the point? And so I'd like to define functional selection as the enrichment or depletion of variants or some sort of proxy of the variants in response to a selective pressure. So you have your DMS library here on the left. You express it in a host, and then some sort of selection occurs, and we'll go over the various types, whether it's a physical selection or within a single pot. You sequence your input in your selected sample, and you end up, you normalize, and you end up with a histogram of variant effect scores that will tell you if your variants are loss of function or wild type like, or some sort of intermediate. There's a ton of factors to consider in these types of approaches. I think a lot of them are actually really specific to the assay that you design, but there's at least a few examples of factors that I think are pretty universal. The first of which is the concept of dynamic range. So just really generally define it as sort of the magnitude of the difference in variant score between your functional and non-functional variants. So I have these nice fake histograms I made here where that sort of are the distribution of variant effect scores for a library. And on the left here, you can see I colored in red a non-functional variant and with 4 measurements of it, and functional in blue. And if you have your distributions of scores are quite overlapping, it's going to be really hard for you to, in a statistically significant way, assign variant scores and distinguish them from one another. So on the right, though, if you have a system where you even have the same level of variation in your measurements, but you actually have a larger dynamic range between your samples, you can more confidently assign variant scores and determine whether something is loss of function or not. Aside from dynamic range, noise is quite prevalent in these assays. Often these are quite long workflows, everything from the library generation phase where you can possibly unbalance your library through overamplification or other methods. When you introduce your library into your host, there's cell-to-cell variability, so you don't have enough cell coverage. Even something like flow sorting for particular approaches, you actually, it can introduce noise in your measurements there. So again, we have this same distribution from the previous slide of the low dynamic range, but in fact, if you actually have a pretty bad dynamic range, but you have less variation in your measurements, you can actually assign variant scores and assign function confidently. Okay, so also functional relevance is important. I think it's really nice to have a goal in mind when you conduct these experiments and to know what you're looking for. Because these different approaches will give you different subsets of information. There's assays, and we'll talk about these more in depth, that will tell you more about a general function of a protein, something like VAMP-seq. Protein abundance is quite relevant for almost every gene. And then there's assays that are more specific to an individual gene's function. and that you have to optimize and select quite carefully for your given gene. And then lastly, I don't want to sound trite by saying throughput and labor is important, but aside from your own sanity, an assay that's easier to perform and iterate on, you can optimize and really improve its performance better. Okay.
Great, thanks, Eric. So I'm next going to go through a few umbrella categories of functional selection methods. And this, of course, is by no means a comprehensive list, and it's going to be largely mammalian cell-based, but we tried to come up with some sort of generic categories under which you can group your thinking as you set out to do your experiment. So we'll cover growth-based assays, assays that use physical separation of cells to phenotype their variants, transcription-based approaches, and then a quick catch-all category at the end. So first we'll talk about growth-based methods. And this is easily the most popular category of mutational scanning and functional selection. It's used across DMSs and CRISPR screens. Has been talked about multiple times today. And again, it's very basic. You essentially start with your normal cells, deliver your variants, let them grow, and compare variance abundance before versus after this cell survival window. Of course, it's most intuitive in phenotypes that say decrease cell proliferation. So say if your variant causes loss of function, the cells bearing those variants are going to die off and be depleted from your final population. A popular application of this assay is to study and phenotype variants in genes that are known to be essential in a cell line, such as BRCA1 in the HAP1 cell line is used by Finley et al. To phenotype that gene. You can also imagine a case in which loss of, say, a tumor suppressor removes the brakes and removes the antiproliferative effects of that wild type protein and then encourages proliferation. So you have enrichment of those variants in your final population. But sometimes your target of interest has no naturally occurring critical function in cell growth, in which case the phenotype of your protein of interest will have to be tied to cell growth by synthetic means, you could say, or environmental means. Examples of this is you could apply some sort of drug selection in which case change of function could confer resistance to your drug. For example, the Lynch syndrome genes can confer resistance to metabolic drugs like 6-thioguanine, an application that's been used by the Kitzman lab at University of Michigan to phenotype those genes. Or if you don't even have a naturally occurring drug resistance phenotype, you could engineer a system as Ethan just described, in which a protein of interest creates a toxicity phenotype or rescues one, as in the case of human Sark, or complementation and rescue of loss of P10 in yeast. So I'll just cover very briefly some pros and cons of this method, this type of method. And of course, there's a reason why it's so popular. It's very easy. If you can do tissue culture, you can do this, and it's relatively cheap. And often there's not a whole lot of setup time. It's often a very clean phenotype. It's very obvious what's going on and relatively easy to develop. And throughput is high. You're limited only by the number of cells that you can culture in passage without bottlenecking your library. It's very simple to do. And the cons, though, of course, are that this is usually going to be specific to your gene of interest, or if it's a synthetic system, it's going to require some sort of setup. So it's a non-generic assay. Also, validating this can potentially be tricky. Is your target gene truly causing the cells to die? A lot of things can cause the cell to die. So it's going to require some sort of orthogonal validation. And it can also be hard to tune extremity of selection. Often the phenotype that you get off the shelf is what you're going to be working with. And this can sometimes be sort of more binary assay. So you can't necessarily tune and optimize the degree of selection you're working with. Okay, that's just the quick summary for growth. Next is what I've called physical separation. For this, rather than waiting for your cells to grow or die off, you're physically separating the cells based on their variants of effects. And by that, I mean, you're literally putting cells into different tubes and fitting them into different Eppendorf tubes based on the variance phenotype. And then within those tubes, you check for the variance abundance. As has been covered, the main example technology used for these sorts of assays is, of course, FACS, fluorescence activated cell sorting, which is, again, physically separating your cells into different tubes. And this has, of course, been used at UW, and now Case Western, in the tech VAMP-seq, where your gene of interest is tethered to a fluorescent protein. So cells can be sorted based on abundance of the protein of interest. Also, instead of tethering, you can also just label your proteins with an antibody and use FACS to sort based on that. Additionally, I know Nick is going to talk about using visual cell sorting to be able to connect a visual phenotype with a cell sorter assisted selection method. And also, beyond using a cell sorter machine, I've seen a common use in literature of using antibody-bound beads to physically capture your cells based on a variance effect of your protein of interest. And technologies used for this are magnetic activated cell sorting, or just the use of IP protocols, such as in Leah's, some of Leah's original work. Okay, and so the main pros of these techniques is often that they are generic. You're either engineering a different fluorescent protein onto your target, or you're swapping out different antibodies, but you're often not starting a protocol from scratch, which is great. And this is huge, especially if your target is not coming with a naturally occurring phenotype. However, as has been described, this is clearly trickier than a growth phenotype, in particular with FACS. I'm just gonna hammer home again, as Kenny and others mentioned, you have to be very wary of the throughput because the number of things in your library is literally gonna be linearly increase the amount of time that you're sitting in front of a FACS machine physically sorting cells. And that can be very painful depending on efficiencies of delivery methods, et cetera. Also figuring out which separation bins, are correlating to an effect can be tricky as well. And that will certainly require setup and optimization. So double check with experts before you pursue this and make sure you have a really robust plan before you sit down at the FACS sorter for the first time. There's a lot of gotchas. And then additionally, at the advantage of genericness comes the cost of specificity and sometimes mechanism. And the methods that that use physical separation tend to be indirect phenotypes rather than something directly related to the original function of your protein. Okay, so that wraps it up for what I have about physical separation methods, and Eric's going to talk about transcription-based methods next.
Great. The idea around transcription-based methods is just that you can link the fitness of your gene or your library to the quantitative expression of some external transcript, the idea, and then you perform RNA-seq and you can look at the relative transcript levels and correlate that back to a variant activity score. This whole approach sort of depends on the concept of a reporter gene. Reporter genes are quite useful and widespread throughout molecular and cell biology. Generally, they exist for a wide variety of genes, but the idea is that you have some sort of exogenous, fluorescent, or luminescent gene, and you tie the expression of such a gene to an external gene's regulatory elements. And so it sort of looks as follows, where you have your gene function, and then downstream of that function will lead to some sort of transcription factor activation. That transcription factor will bind the regulatory region of interest, and then in the case of this example, lead to luciferase production that you can measure the plate reader for luminescence. Now, as is, this approach won't actually be useful for DMS. You can't multiplex luciferase assays. So instead of looking at a luminescent readout, a protein readout, you can actually measure the change in RNA, the transcription of the reporter gene itself. And so if you're doing this one by one, this would involve, just performing qRT-PCR on the transcript. However, in a multiplex setting, you would do RNA-seq. But one thing that's important is since this reporter transcript is not actually your library gene, you need to have some way to segregate which transcripts co-localize with which variants in your library. And this is done through transcript barcoding. People talked about barcoding earlier in this workshop. I won't belabor the point. The idea of which is such here is that we place a short DNA barcode in the UTR of the reporter gene that we link previous to introducing it into the host to the variant library. So in this case here, we have a single variant reporter gene linkage per cell. And then you have your entire library. You lyse it and you prepare the library for RNA-seq and look at the relative expression levels of each barcode. And that is where you correlate to your variant function. So briefly, I'll provide an example. This is actually work I did as a graduate student where we did the deep mutational scan of the beta-2 adrenergic receptor in human cell lines we generated all single amino acid substitutions with oligo arrays, and we tied those variants to the cyclic AMP reporter gene. Cyclic AMP signaling is downstream of G protein signaling, and what we did is we placed a DNA barcode in the 3' UTR of that reporter gene. Then we introduced the library in human cells. The variant receptors expressed at the cell surface, or some of them didn't. We incubated with a known agonist of the receptors, and that triggered G protein signaling, which then, in a manner that was related to the function of the variants, activated the reporter gene. And then we measured the differences in the reporter gene expression with RNA-seq, normalized to the input, and generated variant scores for our entire library. So, you know, like the past two categories, these approaches all have pros and cons. I would say the biggest pro of a transcription-based approach is the relative technical ease. Especially in a mammalian cell context, the cell culture handling is pretty minimal. It's plating, you know, in certain cases applying some sort of selective pressure. You lyse the cells, and then from that point on, preparing a library for amplicon-based RNA-seq is pretty trivial. Use a gene-specific reverse transcriptase primer, and then PCR, and throw it on the sequencer. Another advantage is the modularity of this approach because reporter gene assays exist for many genes. A lot of the concepts and principles of this approach are translatable for these different genes. And you know, you essentially have your gene of interest and you swap a regulatory element out and you have a new assay. So also specificity, because you're measuring a downstream function of the gene, you can really look at something specific. So it really depends on the goal of your experiment, what information you want to get out. Also, you could use this to measure multiple functions as well. One example, at least in GPCRs, they have multiple different G protein signaling pathways, and each of these signaling pathways actually have different response elements. So you could imagine taking the same DMS library and hooking it up to different response elements, and then you can see how these variant libraries actually differentially affect the different signaling outputs. So for the cons, one thing that's super important is this idea of transcriptional noise. So different reporter genes have different basal and induced levels of transcription, and you can imagine if you're doing this in a library setting, you need to accurately sample RNA molecules of your entire library. And so reporters with low outputs, you're going to run into variation in your measurements, and that's going to really inhibit the quality of your results. So it's just important to characterize your reporter gene before you actually run an assay like this. Also, you know, I kind of listed this as a pro in the last slide, but it's a dual-edged sword. It reports on a narrow function. If you want something that's more a holistic approach of the gene, maybe perhaps a gene that you know, leads to cell viability, and you want to look at actual toxicity, you know, maybe that's an approach you would go with. And because it reports on narrow function, you may have to actually run multiple assays to get all the information you want for your given gene. And while a lot of these principles are translatable for different assays, there is a certain level of individual optimization that you're going to have to perform. Whether it's optimizing the growth conditions to make sure you have good dynamic range, the timing of your assay, how long you plate the cells for, and you know, the promoter you use to control the expression of your library, is it too strong? Are you saturating your signal, and so forth. And then.
Great. And then we just wanted to end the transcription-based section by giving a quick shout out to transcriptional readouts in the category of evaluating non-coding mutational scans. So for simplicity, we didn't dive into these, but instead, of course, of using the reporter promoter to read out some cellular signal coming from a protein library. And these are just mutationally scanning, say, the promoter itself or an enhancer of interest or a series of those elements. And these are great, especially for the pros and cons that Eric described, but also because transcriptional output is literally the phenotype by which you want to evaluate regulatory elements. That's all we'll talk about those. So that wraps up the transcriptional readout section. And I'll briefly touch on a catch-all category here at the end. And this is the category where we just put a scattering of things that didn't quite fit in the previous sections, and mainly just to acknowledge the vast amount of functional selection R&D that is ongoing. I'll hop through these, but I do think that the symposium has been a really wonderful way of surveying what's ongoing, what bespoke and specific assays are being developed, as well as ones that are potentially more generic or more deep or multidimensional than stuff just like growth or physical sorting. And in here, in this section, I want to put your target of interest. It may have a specific and unique function that you could adapt to a clean functional selection method. So the sky is the limit, but potentially the sky is a limit. So that kind of leads to the pros and cons of this, and that something's going to be highly specific, which is a huge advantage for assays like these. And it's also going to be really cool and pioneering, which is really exciting. But those are both cons as well. And that it's potentially going to be a lot of R&D and setup to be pioneering it, but also really specific to your function. And so that's our quick survey. And now Eric is going to wrap up before we potentially have discussion and Q&A time or lunch.
Yeah, I just want to briefly summarize. So, we went over a few categories of functional selection methods, and I think that we certainly are missing some stuff, especially from like unique new work that we saw yesterday. The take home is every method has its caveats. There's not a one-size-fits-all solution, so you should really know what the goal of your experiment is before you pick your functional selection method, and really sometimes it's just what's available for you. But yeah, and that's all. So with that, I want to thank you guys and open it up for questions.
So I want to ask about the transcriptional readout, the barcode. It seems like that's pretty critical is having, a barcode to read out the link between your whatever your protein is and your variant. So can you talk a little bit more about like placing the barcode in the 3' UTR or whatever, and then linking the barcode with the variant on the construct?
Sure.
Yeah, I'll take that. So yeah, I didn't.
Question.
Oh, yeah. So the question is, in the case of the transcriptional reporters, how do you actually link the barcode to the gene of interest? So there are a few ways to do it, but the way we did it is so we synthesized the library on oligo arrays. So we actually basically tiled the gene on the arrays and had different backgrounds that we cloned that into. But basically when we cloned it in, the barcode was physically next to the library section. We did barcode mapping with paired end sequencing, and then we basically cloned in the rest of the gene and the reporter gene, which would translocate the barcode into the 3' UTR of the reporter gene so that the construct would be in its final form.
And in your experience, the barcode in UTR doesn't cause any problems as far as?
Well, as far as expression, so yeah, you can always imagine there's going to be some sort of effect on expression, but the way you get around that is you just have barcode redundancy. It's certainly not crazy enough to kill your experiment.
Any more? Yeah.
On that same topic of the transcription-based approaches, you mentioned barcode, but what about linking that to a Western order or self-survival?
So you're saying using the reporter gene, like let's say you can just express GFP or something. Yeah, I mean, that is an option. I would say if you're hook it up to a fluorescent reporter, then you have to sort cells. That's an extra step. I certainly wouldn't want to do that. But it's definitely an approach, yeah.
It also depends, I think, on what phenotype you actually want to read out. So like if you do hook something up to GFP, you're measuring abundance or protein stability or something along those lines, rather than potentially a intracellular signaling effect. Okay, thank you so much for your attention.